Okay, so I thought this would be a nice little opportunity to give you a little bit of the context for how we came to learn about RAS signaling. Because if you think about it compared to some, say, kinases or things like that, the way in which RAS signals is a little bit abstract. It's not this enzyme gets turned on and then it puts a phosphate on something else. It involves a lot of things like conformational changes and recruitment of one component from one place to another. And so it's a little bit abstract, and it's of course easy for me to get up here and tell you what we know, but how did were people able to come to this observation that this GTPase is a signaling molecule and it works through these ways? Because it's not so obvious how you would come to know that. And so the story really begins in the 1960s during the regular passaging of a virus. There were two folks, Harvey and Kirsten, and they were studying these viruses that they found could potently induce cancer, sarcomas, in rats upon infection. So what they were doing here was they were taking rats that had these sarcomas. They would take the sarcomas out and they would break them up into these little chunks of tissue, and they'd grind them up with sand into a paste, basically, and then they would mix this with some buffer and they would pass it through this very fine pore filter. And in this way, this liquid that came out the other end was basically only able to contain things like viruses and stuff. And they would take that filtrate and they would inject it back into newly born rats. And what they found was that when they did this, the rats would develop sarcomas. And so there was something inside these cancers um, that you could collect the filtrate for and re-inject it, and it would produce sarcomas. Okay, so this is the kind of starting observation. And what was interesting was these sarcoma viruses could also transform cell lines. So if they took that filtrate and when they squirted it on some cells in culture, these cells would form foci, basically uncontrolled growth and malignant appearances. And so they reasoned that these viruses that they had isolated must contain something that was able to trigger rapid growth and expansion of cell lines. So what might that be? Well, to find out, they took those cell lines that they transformed and they looked for what proteins were being upregulated or induced in the transformed cells. And what they found was that with these sarcoma viruses, if they infected cells, and then they said, hey, what are these cells? What's getting made when we infect these cells with a virus? They found that their sarcoma virus would lead to the massive induction of a 21 kilodalton protein in the transformed cell. So you can see that right here. They infect with the virus and then boom, the huge induction of this protein. And so they called this protein P21 or RAS for rat sarcoma. Now, what they found, so they figured this protein that's being made probably has something to do with the uncontrolled proliferation of the cells because they don't see that when they infect with other viruses that don't do that. Okay, so what might this protein be? And so they did kind of this very old school experiment where they said, well, let's take extracts that contain this P21 protein, and let's see if something about nucleotide affects the stability of these proteins. And so what they did is they made an extract where they had cells that would upregulate this P21. They would add different things, ATP, TTP, GTP, CTP, to the reaction, and then they would crank up the heat to try to denature these proteins. And their reasoning was that if this protein was interacted with one of these nucleotides, then it would probably be stabilized against heat denaturation more so than if it didn't bind. And interesting, what they found was in extracts in which they incubated GTP into the system, their P21 protein was actually stabilized. So you didn't see that with ATP or TTP, but when GTP was included, the P21 protein was stabilized. And so that suggested to them that P21 was a GTP or GDP binding protein. So this P21 protein must be important. So they decided they would raise an antibody against it. Okay, so they produced, they injected a rabbit or a mouse with this P21 protein, and eventually got this serum that was very good at protecting, uh, uh, at binding to this P21 viral protein. And then they discovered when they were doing experiments uh, to look at this P21 protein in sarcoma transformed lines, in their control lines, these were cells that were uninfected with the virus. Their antibody was actually producing pretty good signal or something in that P21 range, even though these cells weren't infected with virus. So remember that um, the there's not an induction of a P21 protein when they infected with unrelated viruses. But what they're saying here is that there's something in most uninfected cell lines that corresponds to some degree to this P21 protein. And so this suggested to them that there's a normal cellular protein that's related to the viral P21. Okay? And this made them think, okay, there might be a viral protein that's responsible for stimulating uncontrolled growth of cells, and then maybe there's a human natural protein that in a regulated way is responsible for regulating cell growth. And so they set out then to find the cellular counterpart to this viral P21 protein. And this is where the virus ends up being very useful because the genome is super small. And so in what to me seems like a rather heroic set of experiments, they set out to use the viral genome to find a probe that could cross-react with fragments of human DNA that might contain this GTPase. So they took the viral genome, they broke it up into tiny pieces, and they test different radioactive viral fragments to ask, is there a cross-hybridizing fragment here in a digested human genome? And they found indeed that they could produce a small probe that that basically hybridized to something in the human genome that had some correspondence to this P21 protein. And what's great about this is now they have a really good probe that they can use to fish out the cellular P21 sequence. And so to actually find this, what they did was they took genomic DNA from human cells, they digested it with restriction enzymes to generate a library of small fragments that they could easily clone in to a phage. And all they're doing here is nothing weird. They're not trying to use it as a virus for any reason. It's just that each one of these phages is going to get one of these little pieces of human genome. And then they can ask who among these phage contains that P21 protein using the viral derived probe that they had made before. And so they would take these phage, they would plate them out, they'd get these little phage plaques that are basically contain a single one of these fragments. And then they could basically hybridize their radioactive viral probe with each one of these plaques, and then look for the specific plaques that lit up in response to this this viral probe that they had developed. And so basically from this they could find the phage colony that cross-reacted to the probe. And this would basically give them a sequence which they could look at and say, does this look like the P21 protein? And now this is where they did something super old school, which I don't think you'll ever see anybody do anything like this. But they decided they would, once they had this viral P21 sequence and their putative cellular P21 sequence, they stringently annealed those two sequences together and then looked at the annealing reaction by electron microscopy. And what they saw was that they would get these portions of sequence here where the viral P21 and the human P21 would anneal to one another. And so to them, they interpreted this as a region of homology between the viral P21 and the human P21. And with this, they could actually pull out that sequence and get this protein. So what was that human protein actually doing? So they decided to take that human P21 protein and overexpress it in ordinary cell lines. And what they found was that if you massively overexpress this P21 protein, in the same way that the viral protein would induce uncontrolled proliferation and foci formation, so too would the human P21 protein. Okay, so overexpress RAS and cells start forming performing uncontrolled growth, so on and so forth. And additionally, what they started to find was if you looked at the human sequence of RAS in tumors, what they found was that this RAS GTPase, human RAS GTPase sequence often contained mutations. And these were mutations at positions G12, which we'll get into in another lecture, and Q61, which, as we discussed earlier, was one of the residues involved in catalysis. And so now they seemed like they had a collection of observations. There's a human RAS protein that, if you overexpress it, produces foci formation, and in tumors is often associated with mutations. And so from here, what they were able to kind of conclude was that RAS is a human protein whose regulation was affecting decisions about whether to proliferate and grow or not. And from here, the stage was set and kind of the race was on to figure out how it was activated, how it was inactivated, and how activated RAS communicated downstream signals to drive that growth. But until they got to this point, there was really they weren't on the map, had no means of figuring out. But from here, they were able to deduce all of the core logic and regulation that we talked about in the earlier lecture. And so I hope from here you can see kind of this interesting path where we go from knowing nothing to identifying a protein, and then through the process of science, we can arrive at this beautiful textbook explanation for how this protein is regulated. And there's a fantastic read if you're interested in learning more about this history. And I've included a copy of this lecture or copy of this paper on the Canvas website if you're curious to read about it. Again, this isn't something you need to necessarily know for the class, but if you're excited about it or kind of curious about how science works, it's a really interesting history to take in. And so with that, that's all the history I'm going to share. So I will see you Wednesday.